Hello, I'm Peter Mogayzel, and I'm here with Patrick Flume, who is the CF Center Director at the Medical University of South Carolina. We're at the European Cystic Fibrosis meetings in Hamburg, and I'm talking today with Patrick about his studies of VX-770. Patrick, what is VX-770?
So VX-770 is a unique molecule that works on the CF protein, or CFTR. Some CF mutations result in no production of CFTR, and the molecule doesn't get to the cell surface where it belongs and where it works. And other mutations, like G551D, the molecule is made, it gets to where it's supposed to get, but it doesn't work like it's supposed to. And so VX-770 is a potentiator. It acts on that molecule to make it function more appropriately.
And what were the results of your study?
So we did the discovery trial, which was a very important trial with a couple of main goals. We studied the drug in patients with delta F508. The drug was being developed for patients who have the G551D mutation. But this population is less than 5% of the total CF population. So in addition to studying those patients to show that the drug works, you have to study it in more patients to get safety data. So the first and most important outcome of the discovery trial was safety. And I'll just quickly summarize that we studied the drug in patients over 4 months and found no safety issues whatsoever. So that was very promising. But the second goal of the discovery trial was to assess the possibility of some efficacy in patients who have delta F508. Now, in general, we think about delta F508 as one of those mutations in which CFTR is not getting to the cell surface. And so if that's true, then you would not think that VX-770 would have any action, wouldn't work. But in the laboratory, we have evidence that some cells that have delta F508 produce CFTR, and we have evidence that this molecule might actually function or activate that. And so what we wanted to do was to look at patients with delta F508 and see if there were some measures of efficacy. And what we found, generally speaking, was that there was no efficacy. It didn't make any difference in important clinical measures. So for example, in lung function, we didn't see any significant improvement in lung function like was shown in the G551D patients. And those data will be presented at this meeting by Stuart Elborn. Those will be some very exciting results. Interestingly, we did see a small but significant change in sweat chloride, not large enough to be of any clinical relevance, but it does give us a hint that maybe just like in the laboratory, there are some patients who get delta F508 CFTR to the cell surface and this drug may act on it.
So there may in fact be a role for VX-770 in patients with delta F508 looking forward with these future studies.
As a monotherapy, no. The evidence to us in this study is that it's not going to be a drug that can be used alone. But if it was used in combination with some other product, a corrector, that can increase the amount of CFTR getting to the cell surface, then it suggests there might be some hope for this drug in those patients.
And trials of that combination therapy are ongoing now, aren't they?
There are trials going on in combination with a corrector. The company that produced VX-770, or Vertex, has another compound, 809, which functions as a corrector. So we're in the early stages of investigation looking at the combination of 809 and 770. 809 to increase the amount of CFTR and then 770 to act on it. But this is very early stage development. We're just getting into dose finding to figure out where we might go forward with future clinical trials.
And what about the use of VX-770 with other mutations?
Well, the major question that everyone's going to ask is, will this work for my child or in my case? And so here we talk about G551D mutations in whom the studies were done and look very favorable, and delta F508 where it looks like that's not the case, but everyone is going to be asking about other mutations. And the short answer to that is we don't know. And I'd like to be able to tell you that we could do a simple test, take the medicine and measure your sweat chloride, and that would tell you whether the drug should be working or not. But I don't think we have the data yet to support that kind of surrogate marker to know if the drug is working.
No, but that's a very intriguing idea because it would allow you to differentiate the patients that would really respond to the drug from those that wouldn't.
Oh, it'd be fabulous. You know, you're always looking for some other marker, a surrogate, that if I could measure this and demonstrate that it's working, then it will have eventually clinical benefit in my patient. And that's what we really care about, is the clinical benefit. But that's going to require more work to tease out whether there's some relationship between the change in sweat chloride and other clinical measures, or whether there needs to be some threshold response, that you need to see a change of this much in your sweat chloride, and then you could predict a clinical outcome.
All right. Well, great. Thank you very much, Patrick.
Oh, my pleasure.